en Israël. Donc Tawfik a fait son postdoc à Cambridge chez Ferch, on va dire qui est une sommité dans l'enzymologie. Et son travail actuel dans son équipe Weizmann, c'est de trouver l'évolution, comment les enzymes obtiennent de nouvelles fonctions. Donc c'est justement très complémentaire justement avec le travail de beaucoup parmi vous qui sont justement de trouver l'évolution avec la phylogénie, et ça donne le pendant. Et justement vous allez voir aussi que la passion de Dan qui nous expose ici, qui est l'escalade, lui a permis de beaucoup réfléchir et inspirer justement pour ses travaux prestigieux dans ce domaine d'évolution des enzymes.
Merci and thank you very much for the invitation to be here. It's, coming to France is always a pleasure, and now looking back to my career, I cannot understand how I was able to spend 6 years in Cambridge that is flat and there's also English weather and English food. So this is why I enjoy coming to France. And as Eric mentioned, I have two obsessions in life. One is mountains. So the talk is punctuated with photos from various places in the world that I visit. Some are, the desert ones are from Wadi Rum in Jordan, but there are also various places in Europe, including 2 days ago I was in the Gorge de Verdon here in France. So the other obsession I have is regards proteins and especially enzymes. So I probably don't have to convince you how fantastic are the performances of proteins in general and enzymes in particular. But just to illustrate it, I've depicted here data regarding several key biochemical transformations that are essential for life. And in blue here are the rates of the spontaneous uncatalyzed reaction at 37 degrees. And you can see that these reactions that are essential for life would occur with a half-life that may correspond to anything from 1 minute, this is hydration of carbon dioxide, to 1 billion years. This is the rate of spontaneous hydrolysis of the phosphodiester bonds in DNA. What enzymes do is to accelerate the rate of these transformations such that they occur on a time scale. So here is the plotted like the half-life of the enzymatic reaction of between a minute and a microsecond. In doing so, the rates of some of these transformations are accelerated by 17 orders of magnitude. This means by analogy that by the time I reach the door, the enzyme will be in Pluto at the end of our solar system. So this is the kind of things that proteins can do. On top of catalytic efficiency, enzymes and other proteins we will discuss also show exquisite specificity. They will bind their substrate but not other substrates. And of course we ascribe this performance to the struggle for survival, for selection, acting on biological entities for a long period of time to enable them to meet various environmental changes and win this competition for survival. At the protein structure function level, the way we would normally explain at the level of a textbook why proteins are so proficient in what they do and how they achieve exquisite specificity, it relates to this model, the famous lock and key model by Emil Fischer, which is now not Emil Fischer, but the model is well over 100 years ago. And by this model, we have this perfect complementarity between the protein binding or active site and its natural substrate or ligand. And this perfect complementarity accounts for the efficiency and specificity of proteins. So in terms of explaining how proteins work, this model is perfectly suitable, of course. And also implicit in this model, something we will discuss later, is this idea that if we have one sequence, one gene sequence that encodes a particular one sequence of a protein, this also means one three-dimensional structure, or maybe few 3D structures that are related to the function of the protein and one function. So I will start with an example of how evolution shaped the exquisite specificity of a given protein. And the examples I've shown today relate more to my French connection, if you like, to the long-standing links with the Eric's lab and to the work that Michael Elias has done in my laboratory. So we will discuss the issue of exquisite specificity in relation for the struggle of organisms, and specifically microorganisms, for phosphate. So phosphate is, of course, absolutely essential for life. And we will see an attempt to break this paradigm in a moment. However, it is not very abundant on this planet. So here is a diagram of the main elements on the Earth's crust. And phosphorus is in this point, amongst these 0.14% of other elements. So it's a relatively rare element. Moreover, unlike carbon, oxygen, hydrogen, and nitrogen that are available as gases, phosphate is not. It's in minerals. Most of these minerals are inaccessible also to organisms. So there is definitely a struggle or a strong competition for phosphate. And given this background, a work that was published about 3 years ago in Science by a group by Felisa Wolf-Simon that works in a NASA laboratory created a lot of attention, or drew a lot of attention. And Felisa Wolf-Simon described an enzyme, described a bacterium isolated from a lake in California called Mono Lake, where the arsenate concentration is very high. The claim she made is not only that this bacterium can survive these high arsenate concentrations. This was not new. People have isolated before bacteria that live in high arsenate environment, but she went a step further to claim that this bacterium can grow using arsenate instead of phosphate. And this claim has drawn a lot of antagonistic responses for very good reasons. So these are coverage soon after the paper was published, like the arsenic-based life aftermath and the microbe gets toxic responses, because thousands of biochemists thought that this idea was complete stupidity for reasons that we will discuss in a couple of moments. But what actually brought Wolf-Simon to this, you may say, ridiculous proposition that arsenate can take over phosphate? The underlying reason is that these two anions are extremely similar in their physical and chemical properties. So what you see here is like the two ions superposed, and it's very hard to distinguish them between them. The reason being that their diameters differ by only 4%, by 0.04 angstrom. They have identical valency, so it's also arsenate O4-3 shown here. Identical pKa values and very similar distribution of the negative charge. So why why not arsenate? Why did, and actually another point of course is that arsenate is as abundant or even more abundant in certain respects than phosphate. So why did nature choose phosphate, or why did nature did not choose arsenate? Turns out that this is not a new question. Actually the first enzymologists, if you like, have dealt with this idea, starting with Otto Warburg, the scientist that underlined our first steps of understanding metabolism, and going to Dixon and very highly distinguished enzymologists, have found out two interesting points. The first one is that enzymes that use phosphate are unable to distinguish between the two anions. So they would take arsenate and use it instead, thus creating, for example, if you look at this enzyme, aspartate dehydrogenase, that takes phosphate, so it's oxidative phosphorylation to convert this aldehyde into a phosphoester. If you give this enzyme arsenate, it will do exactly the same. The difference, however, is that whereas this is relatively stable, the arsenic or the arsenate ester will hydrolyze in microseconds. Similarly, if you were to create DNA with arsenate instead of phosphate in the diester bonds, it would break down within minutes instead of billion years, as we discussed in the first slide. So this is the primary reason that life is based on phosphate and not on arsenate. The instability of arsenic esters like this one. And this is also the basis of using arsenate to decouple metabolism, because the metabolism would create these esters, investing ATP and other energy means, but then would burn them in vain because they're hydrolyzed immediately. However, what's also clear, or was clear in the literature at the time I looked at these papers after the publication of the arsenic paper, was that there was no evidence whatsoever that proteins can actually distinguish between these two anions. So there are about half a dozen crystal structures actually of enzymes that utilize phosphate, including the dehydrogenase we discussed before, and crystallize both with phosphate and arsenate, and they superpose perfectly. The interactions, the binding residues, even the water molecules are placed exactly the same. This is in contrast to ions that are slightly different. Even iotate would already bind in a different mode, and then you can see some rearrangements of the binding residues. So it seems that proteins can distinguish between different anions, but not between arsenate and phosphate. This is another crystal structure of another enzyme, exactly the same picture. So at this point, I came to a few people in the lab, including Mikael, and said, okay, whilst it's clear that the claim that this bacterium uses arsenate is like, can be disregarded, the question how this bacterium survives in this environment that is rich in arsenate, despite the fact that its proteins are unable to distinguish it from phosphate, is still a valid question. And this is where Mikael has proposed to look to examine a system that he has worked on during his PhD with Eric, and that is the phosphate-specific transport system, or the PSTs. So this system that is universal in all microorganisms, in all bacteria, is comprised of an ABC transporter, ATP-driven transporter, and the binding protein that is periplasmic or on the outer membrane. And the mechanism is such that the periplasmic binding protein will capture the cargo, phosphate anions in this case, and would then hand it over to the transporter that would push it into the cells. And turns out that this family of phosphate binding proteins has been characterized in detail and has been studied extensively in the past, including its selectivity, but the focus has been on how sulfate and phosphate are discriminated, and sulfate is a very different ion in charge and valency than phosphate. And it has not been examined whether at all these proteins can distinguish arsenate and how they would do it. So what Michael and Alon, a student in the lab, has done is to take 3 different phosphate-binding proteins from these 3 different bacteria. Some from arsenate-sensitive species that have not been exposed extensively to arsenate and have been reported to be sensitive. And these are E. coli and Pseudomonas fluorescens. And then some arsenate-resistant bacteria isolated from lakes like monolake that contain arsenate. And in addition, Michael and Alon have cloned two PSTS, 2 phosphate binding proteins that have been identified in the genome of this bacterium from monolake, this GFAJ1 strain. And then they tested in a discrimination assay where you test the displacement of a phosphate labeled with P32 with arsenate. So what you see here is percent of replacement versus the excess of arsenate over phosphate. And what they found is that those phosphate binding proteins from bacteria that are sensitive or somewhat resistant to arsenate distinguish by about or nearly 1,000-fold against arsenate. So this was actually the first indication that proteins can distinguish and significantly distinguish between these 2 anions. The bacterium from monolake, the scandal bacterium, has 2 phosphate binding proteins that we discussed. One of them behaves exactly like the E. coli and the Pseudomonas fluorescens. But one of them, that we dubbed PVP2, distinguishes by another 10-fold to give you nearly 10,000-fold discrimination. And with some colleagues at ETH, we could see that this isoprotein, the PVP2, is turned on, transcription of this protein is turned on upon phosphate starvation. So it was clear that this protein relates to the physiology under limiting conditions for phosphate. So the answer from these studies has been that yes, proteins can distinguish between these 2 anions, but it did not reveal what would be the mechanism. How would 2 very similar anions be bound in a different way? So to answer this question, Michael has taken the protein, the fluidine, the Pseudomonas PVP that he's worked on in PhD, and where together with Eric, they solved the crystal structure with phosphate. And they've also solved the crystal structure of the same protein bound with arsenic. The great thing about this protein is that one can get sub-angstrom resolution in the crystal structures, which allows you to see the hydrogens, as you see in this density map. And this has turned out to be crucial for understanding how this protein distinguishes. Because, very embarrassingly, when Michael came with a crystal structure of the arsenate and superposed it on the phosphate, it looked exactly the same. So these are the 2 complexes, superposition. These are the 2 anions. You can see they are exactly in the same place. So are the oxygens, and so are the ligating residues. And again, I would emphasize that this is a sum-angstrom resolution, so the error here of determination of the location of the atoms is very, very small. So essentially just looking at the structure as is tells you nothing because it looks exactly the same. However, upon going one interaction by another, and there are 12 hydrogen bonds here, it's a very extensive network that of the protein binding the anions, Mikhail noticed a very special bond, this hydrogen bond with aspartic 62. And when you examine this bond in the complex with phosphate, you see that you can also identify some density for the hydrogen. You see that the bond angles are taken from the textbook. This is like 122 degrees, like you would see very close to water. This OHO is a straight line. It's like very close to 180 degrees. And likewise, this angle is 109 degrees. With arsenate, moreover, the hydrogen is located almost in the middle between the two oxygens, suggesting that this interaction is energetically intense because it's close to a covalent bond. However, with arsenate, the bonds look completely freaky. This is 162 degrees. This angle of 127 is not seen in any hydrogen bond, even small molecules, and likewise 95 degrees. So essentially nature has evolved a pocket that is able to distill this very small difference of 4% in size into a very significant difference with respect to a single hydrogen bond. Indeed, mutating this aspartic to asparagine results in a loss of about 10-fold in the discrimination. Further, we could see that the anions are bound extremely tightly. There are van der Waals even clashes, I would say, with binding site residues that result in these anions being bound very tightly and unable to move even a fraction of an angstrom. And this is the reason why phosphate and arsenate are actually bound in exactly the same coordinates. So this seems to be like a perfectly good and convincing example for this model of Emil Fischer, the log and the key, where there is perfect complementarity for phosphate and the wrong key, in this case arsenate, is unable to open, or you would demand 1,000-fold higher concentration in order to go into the active site. And therefore this model is perfectly suitable to explain how proteins perform their function with this exquisite capabilities. What this model is less good or inadequate, even I would argue, to explain is the following dichotomy, that we know that whilst proteins show this very high proficiency and exquisite specificity, they are also highly evolvable. That means that upon change in the environment, they can change their function and specificity. And of course, these two properties are contradictory, not in one way, but in many ways. Every engineer will tell you that if you want high performance, you need to design and build a machine that specializes in doing one thing. And it will do it very well, but will show zero versatility. If you need to modify it, it will be impossible because it has been designed for one purpose. With proteins, we experience this dichotomy that they are extremely well designed, if you like, for what they do, as we've seen with the phosphate and arsenate, or these enzymes that I discussed as examples. But on the other hand, they are also highly evolvable and able to change. So how does nature bridge these two demanding requirements? So I've chosen another example that relates again for this struggle, and specifically of soil bacteria to obtain phosphate. And the example revolves around an enzyme that is also studied, has been studied in Eric's lab. It is a bacterial enzyme called PTE, or phosphotriesterase, that has been isolated or found on a plasmid in soil bacteria. And no natural substrates, metabolites have been identified for this enzyme. But on the other hand, this molecule called paraoxone or parathione, we have a sulfur here, comprises an excellent substrate for this enzyme. In fact, the K-cut-over-K catalytic efficiency towards this substrate is amongst the top 5% of enzymes. Only 5% of all enzymes get to this catalytic performance. And many structure function studies have indicated that this is the substrate for this enzyme. The snag is that this compound has been first introduced in the mid-50s, and the enzyme was already cloned and isolated in the 80s, mid-80s. So it seems that within 30 years, soil bacteria have been able to evolve an enzyme that is amongst the best enzymes for a compound that has never ever been on this planet until the mid-50s. Why? It's quite clear that in response to phosphate deficiency, this organophosphate can be turned into a source of phosphate by hydrolyzing these bonds, and indeed it was found that Pseudomonas strains that carry this enzyme can grow with this pesticide as the only phosphorus source. Moreover, this trick of evolving an enzyme overnight is not once in a lifetime show. Turns out that similar enzymes were isolated around the planet, in the US, in Australia, and also in China, from an enzyme that has no sequence or structural relationships to the other two. So it's a trick that nature can pull not only once, but repeatedly. So how did it happen? How within less than 50 years, nature was able to get this highly proficient enzyme that degrades a xenobiotic that is new to this plant? This is where this model stops to deliver insights, and where we might consider an alternative model that I will present as we go along. So, Livnat Afriyat, a PhD student in my lab, took this project of trying to understand the evolutionary history of PTE, of this enzyme. So the first thing you do is, of course, look for relationships, for uncles, aunts, cousins of this enzyme. And indeed, you can identify several families that are related in sequence. And even there were crystal structures at the time, so that they looked nearly identical. These families have exactly the same fold and exactly the same active site arrangement that is bimetallocentric. However, what these families do was unknown at the time. Further, the sequence identity between these PTEs and the closest enzymes were only 30%. So despite the structures being identical, the sequence was diverged by over 70%. So how would you figure out what these enzymes are doing? Interestingly enough, they were identified to have weak paraoxonase activity. And this began to make sense because since kind of the late 90s, I've been advocating for the following model to replace or update, if you like, the Emil Fischer lock and key. By this model, we have the native conformation that obeys the model of the lock and key of Fischer. However, proteins exist actually as an ensemble of many conformations that are in equilibrium with each other, with very fast exchanges. And with the idea that some of these alternative conformations may also mediate alternative promiscuous functions. That is to say, functions that this protein has not evolved for and are not even appeared throughout its evolutionary history. They are coincidental, fortuitous, promiscuous. They have not been shaped by evolution. They are just an outcome of, if you like, the promiscuity of structures of proteins and the availability of these alternative conformations that can turn into alternative active sites, if you will. And I will not go into our early demonstrations where we could show, let's say, an antibody could adopt two conformations, each binding a completely different ligand and so on, but we'll focus on more recent results, yes. And they are related to what you may call enzyme promiscuity, this idea that a single enzyme can catalyze not only the native, the original reaction for which it's evolved and that is used normally in the physiology of the organism, but can have a whole array of coincidental promiscuous reactions that it can catalyze with no relation to the physiology. And this was actually a very important hint regarding also evolutionary relationship, because the rationale we followed is we said, okay, if these families that are evolutionary related have weak, probably promiscuous para-oxonase activity, some weak activities that the para-oxonase may have may actually be the original or the native activity of these families, with the idea that if they share a common origin, as we will see in a moment, there would be also some overlap that relates to the evolutionary history, meaning the promiscuous activity of the para-oxonase would be a vestige of what its ancestor was doing before it diverged. And then by combination of this, applying this rationale and some lab, Livnat-Afriyat tried some lactose as substrates, and turned out that the PTE that is highly proficient in hydrolyzing para-oxon was also showing weak activity with lactose, these cyclic esters. And this allowed them to characterize this family, and to cut a long story short, it turned out that this family specializes in these substrates that are called homoserine lactones that are used as quorum sensing molecules in bacteria. And that members of this family, including SSO-CoX that was characterized in Eric's lab, show weak para-oxonase activity as a promiscuous, coincidental activity, because this family evolved well over 100 million years ago, before this planet has seen us, let alone the chemicals that we spray over. And conversely, what is the main activity, the native activity of this family, lactonases, and specifically hydrolyzing this type of lactones, is a weak promiscuous activity of the PTEs. It's a vestige, a relic of what this enzyme was doing before it got exposed to para-oxon and diverged into a proficient para-oxonase. And indeed, this mysterious overlap between lactase and phosphophoesphorases has been found to be an underlying theme in all the families that evolved into para-oxonases. They are all stem from lactonases, and they all show, as a structural study done by Michael, this overlap between para-oxon in blue and lactones here in purple, regardless of the fact that they are completely different folds and different catalytic machinery. However, this overlap between the structural overlap between these two substrates, one being the native, one being the coincidental, is not the whole story. Turns out that according to the scheme that I presented in the beginning, in one of the enzymes, that this is a human lactonase that we analyzed that also has promiscuous peroxidase activity, a different active site configuration is used for both activity. So this is actually a crystal structure. This shows the envelope of the active site in a crystal structure where a lactone is bound to the enzyme. And this is peroxone superposed into this structure, and you see that it breaks outside the active site wall. So basically the configuration of the enzyme that binds and hydrolyzes lactone is unable to bind peroxone. There is a significant rearrangement of an active site loop going from this blue with a bound lactone to this gold depicted configuration that opens the active site, and in this alternative configuration peroxone is bound and hydrolyzed. Even further, some modeling and crystal structures we have done have shown that the mode of binding and location of the catalytic calcium differs between the native activity, this is the way, the place where the calcium is for lactone hydrolysis, versus peroxonase where the calcium dislocates 1.5 angstrom upwards to catalyze the promiscuous peroxonase activity. And this is quite unprecedented because your metal binding sites are extremely conserved in enzymes. So we have cases here of one enzyme doing multiple reactions by virtue of having multiple configurations of the active site, movement of loops, movements of chain chains, movements of catalytic metals, etc., etc. And this principle has been demonstrated by my lab and now by many other labs in a variety of cases. I will not go into too much detail, but this is, for example, a study of Kalia and Levin on how new protein-protein interactions evolve. And again, you could see the same principle, where in this time it's two different binding configurations of the binding partner. Two is in acid. Two different configurations shown in blue versus the yellow that are used in order to develop a new binding partner. So again, the ability here of proteins to bind in a different configuration is the basis of a specificity switch in this family, which are DNases and their binding inhibitors. And we have even described more extreme cases of the scaffold, the very fold of the protein changing. So this is an evolutionary transition whereby from a monomeric to a pentameric protein, where the very same sequence, so these polypeptide chains that have a very different configuration of fold as you see here, are actually exactly the same sequence. Rearranged in 3 different structures to give you this pentamer, which is a homopentamer sequence, but a heteropentamer structure. So the bottom line from what I've shown here is to try to convince you that this model of proteins coexisting in many conformations and some alternative conformations providing this promiscuous function explains probably how nature found a starting point that has some low level of activity to hydrolyze a new compound, peroxone, that appeared on the planet, and therefore provide the initiation for a new enzyme. However, these promiscuous activities are typically million-fold less developed than the native activity. So how does a starting point that is coincidental and weak develops into a fully fledged, highly reactive enzyme? This is where the main, if you like, expertise of the lab kicks in, because what you really like to do is to understand the evolutionary processes, not just by looking at the starting point and the end point. In our case, the end point is PTE, the paroxysmal hydrolyzing enzyme, and the starting point would be a lactonase with a promiscuous peroxidase activity, but also actually understand the entire pathway, the trajectory, the intermediates that link the starting and the end point. And these are long gone and disappeared, and the likelihood that you will find them walking around is zero. So this is where this idea comes in of trying to reproduce the evolutionary process in the laboratory in order to isolate various intermediates along this trajectory. In the case of PTE, this has proven very challenging because the two enzyme families are separated by 70% sequence divergence, which means about 200 amino acid replacements. And more complicated, they are also separated by the backbone by certain loops in the active site, being in this case much longer in the newly evolved enzyme relative to the progenitors. And structural studies done in several labs have indicated that the changes in the loop configuration are actually a key to the new specificity. And in particular, Lignat identified this insertion of 8 amino acids in loop that is called loop 7 within the active site of the new enzyme of PTE relative to the lactonases. And once we've had these alignments that are quite difficult to obtain, she could go to the PTE and try to truncate this loop, to cut this insertion that probably occurred sometime between 50 and 30 years ago. And this was found to be the trick in reconstructing the ancestor of PTE. Because upon deleting this insertion in loop 7, together with a single point mutation in a residue not far from the loop, the following has happened. So this is the wild type peroxonase. As you can see here in terms of K-cut/Km, it's an extremely efficient enzyme, 40 million with peroxone. It has some lactonase activity with artificial chromogenic lactones, but no detectable activity with homoserine lactones, with the quorum sensing model. Upon truncating and putting this point mutation, the enzyme lost about 30-fold in the peroxonase activity, but it's still a highly reactive enzyme, as you see here. The K-cut/Km exceeds 1 million. It's a very respectable peroxonase still. But by now, the ability to hydrolyze these homoserine lactones here. And actually, the enzyme is not a bad homoserine lactonase. Its homoserine lactonase compares within an order of magnitude to this enzyme family that we discussed before that PTE comes from. So in fact, what we have obtained here is a bifunctional ancestor. And unlike the two specialized families that have only peroxonase or primarily peroxonase and primarily lactonase activity, this progenitor can do both. And in general, we think that this idea of moving from specialists, enzymes that do, or proteins that do one thing, into generalists, enzymes that are bifunctional, and back to specialists, is a general theme in the evolution of organisms as well as proteins. And to illustrate this, I will give you one example that again stems from this laboratory evolution experiment, where Nobuto Kariki, another very talented postdoc in the lab, performed this long trajectory of 18 rounds of random mutagenesis and selection to convert the PTE that we've discussed into an enzyme that is an aryl esterase that cleaves carboxyesters instead of phosphate. And in this, it described the first trajectory of an enzyme respecializing in new activity. And what we had experienced followed exactly this principle that we discussed before of moving initially from a specialist to the generalist. So 6 rounds of evolution, it produced 6 mutations that increased the new activity under selection of the aryl esterase by over 1,000-fold, but only to sacrifice 15-fold of the original activity. So the ratio of gain to loss was over 200, meaning very weak trade-off, moving to an enzyme that is bifunctional, that is a very good paraoxonase and a moderately good aryl esterase. This trend, however, reverted as Novo continued to hammer in rounds of mutation and selection to move to a new specialist. Now, the gains per round were very small. And in order to gain another just 10-fold in the new evolving activity, 700-fold of the original activity was sacrificed, and the trade-off became very large. That is to say, to gain a bit, we had to lose it, or the enzyme had to lose a lot. So we have 2 phases, we believe, in the evolution of new functions, going from a specialist to a generalist and then again to a specialist. Lastly, I would like to tell you in 5 minutes about a bit more the practical things that the lab does. So thus far we've discussed the fundamental aspects of protein evolution, but just to give you some impression that all these ideas that are very theoretical about promiscuity and generalist and multiple conformations and multiple functions and so on, this knowledge, this theoretical knowledge can be also translated into very powerful enzyme engineering in the laboratory, using again this algorithm of performing evolution in the laboratory. These cycles of creating genetic diversity and putting it under selection for a new function. And again, following the examples we have already discussed, my lab has been doing some work to engineer enzymes for the detoxification of pesticides. So we discussed paraoxone, but also of other type of compounds that are very similar. These are organic phosphates that were designed, actually the first one, this one, the first 2 were designed by the same German chemist that designed paraoxone, but to inhibit not insect acetylcholinesterase, but human acetylcholinesterase, and to kill. And these are nerve agents from various types. So these being like xenobiotic artificial chemicals, no enzyme has evolved to degrade these compounds. So this is where this idea of promiscuity and alternative reactions and conformation kicks in, where we can identify starting points that have the initial chemical reactivity to hydrolyze these nasty compounds and use them as the basis for evolutionary optimization for getting highly efficient enzymes. So I'll just give you some highlights from this study. This is the work of Rinko Devi Gupta with Moshe Goldsmith that took a human lactonase the activity of which with the toxic isomer of nerve agents such as cycloserine is extremely low. This is the K cut over Km. And amplified it by over 4 orders of magnitude to get an enzyme with a K cut over Km in molar -1, minute -1 that is well over 10 to the 7. The power of this evolutionary algorithm, if you like, is also manifested in the changes in stereospecificity. So actually the wild-type enzymes, unfortunately, favors the non-toxic isomer. These compounds are enantiomeric with respect to the phosphate. It favors the non-toxic isomer by over 1,000-fold. This is the activity with the S, this is the activity with the R, this is the S over R ratio. By the time the evolutionary process reached this stage, the stereospecificity was completely reverted, such that the S isomer is favored by well over 1,000-fold, giving you a switch of 1,000,000-fold in selectivity. More recently, we've done the same trick with the bacterial PTE with V agents, and V agents are chemicals of this structure. They are the nastiest, most toxic nerve agents. There is currently no enzyme that can degrade the toxic isomer with efficiency that is even close to what you would need for detoxification in vivo. In collaboration with David Baker's group and Claire Brisson that did computational design, we could improve by thousands of folds the activity towards the toxic isomer and even engineer a specific pocket that coordinates this very special choline group that is borrowed from acetylcholine. The nerve agents were designed to mimic acetylcholine, of course, to get a highly specific pocket that binds the choline. So just to conclude, I'd like to leave you maybe with one slide you may remember at best, and this is this idea or this the protein dichotomy. That side-by-side proteins are able to do one task, they are non-native tasks with extremely high efficiency and specificity, but they also maintain the ability to evolve and change very rapidly. And these are encompassed in this scheme that on the one hand has the native conformer that obeys the lock and key model, but the alternative conformations open the door for alternative functions as explained the evolvability of proteins. And lastly, I would like to again mention the individuals that do this. All I do these days is to travel from one nice place to another, meet interesting people and climb nice walks. This is Dolomites in Italy. But the people that actually did the work are Alon Werner and Michael Elias, whose work on the PDPs are discussed. Livnat Afriyat, who invested several years in tracking down the evolutionary history of the PTE, and lastly Novoto Kariki with a long evolutionary trajectory. And thank you for your attention.
Des questions?
Yeah. But you didn't convince us that the evolution of your enzymes did take place during the last 50 years. To increase the catalytic rate, there are two different ways to do this. We can either evolve the enzyme, or we can evolve the substrate. And here you're facing the enzyme that was pre-existing actually with a new substrate that just happens to be more reactive than a natural substrate. Am I correct?
But how would you evolve?
This is well known. There are thousands of other enzymes where you can make a man-made substrate that will react faster than the natural enzyme.
With this I agree. But the issue is that, I mean,
in that case, of
the selection,
then a simple phylogenetic tree should be able to trace back when the evolutionary event took place.
But the challenge is given. I mean, this pesticide appeared as an alternative phosphate source.
Sure. But that pesticide may just be a good substrate.
I think that, I mean, and I can show you results from experiments we've done, that basically if you take not just an unnatural substrate, but an unnatural reaction, a reaction of a type that we have no precedent when an enzyme can catalyze, and you encounter it with a given protein, say the protein of E. coli, you would easily identify enzymes that would tackle this unnatural substrate and unnatural reaction with low efficiency. So what I think is that
I agree with the principles, but what you say on the principles is absolutely right and correct, and well documented. In this particular case here, you cannot exclude that the substrate is just a better substrate than what is the real substrate. And we just don't know what the real substrate is, if I understand what you said.
Which
real
one?
Substrate for peroxidase?
No, it seems from, you cannot prove this, but it seems from the catalytic performance, because this type of K cut over Km, only 5% of enzymes get to this level with their natural substrate. But also if you look at various structural parameters and so on, it seems that this enzyme is tailored for peroxide. This doesn't mean that there isn't, some likelihood that one day someone will find another substrate.
For the medical people here, it should be clear that the fastest substrate is not necessarily the natural substrate.
With that I agree.
Should be very clear.
Yeah, and it's the other point is that I don't know, but it's the same question I wrote a long time ago with antibiotics. And what we found, and of course, at the beginning we were thinking that it was an adaptation of the modification of pre-existing enzymes that have been selected and the suppression of the antibiotics. But now, basically when we look at very old samples, we find that the enzymes that destroy the antibiotics were there thousands of years before they have been in contact with the modern enzyme, modern antibiotics. That means that the enzymes were there in a way or another doing something else that maybe we don't know.
Yeah.
It's the way efficient.
No, with that I fully agree, because as I mentioned, these lactamases have been probably around for 100 million years. That is what we see from the phylogeny. And then nature capitalized on I agree. I mean, this is the paradox of evolution. Nothing evolves unless it already exists to some degree. So it's the same with antibiotics.
I'm not sure I agree with it either. I think that there is still creation of new products. I mean, this has been illustrated. This has been
but not, I mean, evolution is about, this slide I removed, but I always explain it's like climbing to find the line of least resistance. You don't go where you kill yourself. You find some line that, so it's a question of availability. If there is a protein that can do the job suboptimally and can be optimized, fine. If not, I'm not excluding, and there are probably some few precedents for the de novo creation of even human proteins.
So there is plenty of evidence of new creation of proteins that have been efficient. I'm sure you remember this paper on the first position in Nature on Drosophila, where they look at orphan genes. So the orphan genes are neo-created proteins.
Yes, we can discuss it later, but not I think many orphanages are because we haven't found their family members, but not. I think with the enzymes, I mean, what we see where there is clear-cut evidence for the de novo creation of the protein, it's typically these disordered proteins that are involved in regulation. And I at least don't know an example for an enzyme, simply because you would have to create something that is.
Yeah, but it's hard to believe that. Personally, I cannot believe that all the orphan proteins are just proteins that were around since 4 billion years, and that you get all these proteins since 4 billion years. This is impossible. It's just, I mean, nature is testing all the time, degradation enzyme, genetic sequence, fusioning, and some of these by chance are new proteins, and they may be efficient. And this is creation all the time. And the fact is that whatever you're doing in the genetic era, I mean, at each time you sequence something, you get new orphan. You don't get less, you get new orphan and new orphan. So the number of orphan is still growing like that. It means that it will never resolve. I mean, it's creation.
Not creation, evolution.
That's creation. Well, evolution at the base level, not at the protein level.
I think
well, yes, that's
disagree.
That's it.
No, I think, but we probably don't disagree that between the origin of life and now there is a big time span. So I don't think that all these orphan genes are recently emerged like some of the enzymes we discussed here, or like some of the new antibiotic resistance. They could have been created at some point between 4 billion years ago and now.
Yeah, but if they could have been created, that means that there is protein created all the time.
Yeah.
D'autres questions? Merci beaucoup.